We'll leave the cell wall synthesis inhibitors now, and we'll talk about some other types of antibiotic compounds. These are produced not by fungal cells, but by bacterial cells. Remember, the reason that a bacterium would want to produce an antibiotic is so it can fight off other bacteria that might want to harm it. So these antibiotics fall into the category of translation inhibitors, and they include classes called aminoglycosides, macrolides, and tetracyclines. Let's start with the aminoglycosides. These are antibiotics that target the small subunit of the ribosome, or again, what's known as the 30S subunit of the ribosome. These drugs bind to and interfere with this subunit, and that prevents the ribosome from assembling and doing its job, which is performing translation. So these drugs are inhibiting translation and therefore inhibiting protein synthesis. Aminoglycosides are narrow spectrum. They are effective against gram-negative bacteria. And they include drugs like gentamicin and amikacin and streptomycin and so on. The macrolides are also translation inhibitors, but they target the large subunit of the ribosome. So they bind to the large subunit and they prevent the ribosome from assembling and doing its job. The macrolides are actually broad spectrum. They are effective against both gram-positive and gram-negative bacteria. And they include drugs like erythromycin, azithromycin, clarithromycin, and so on. And finally, there's the tetracyclines. Tetracyclines target the small subunit, so they're similar to aminoglycosides, but they are broad spectrum, effective against both gram-positive and gram-negative bacteria. There are lots of different types of tetracycline. In other words, the tetracycline molecule has been modified quite a bit in the laboratory to produce drugs like chlorotetracycline and oxytetracycline. There are several different semisynthetics available for this drug. I just want to take a minute to note that tetracycline is actually an old drug. And for many, many years, tetracycline was used as a feed additive in livestock. And the reason it was used was because it was discovered that a side effect of low doses of tetracycline is growth promotion. And what I mean by that is animals that are fed low doses of tetracycline in their grain every day grow more quickly than animals who don't get tetracycline. It's important to know that when tetracycline was used as a feed additive, it was given to the animals at a low dose. It wasn't the dose that would kill bacteria. It was lower than that. So when you hear that antibiotics in animal feed contributed to the resistance of bacteria, it's not correct. No bacteria were being killed by this amount of tetracycline in these animals. It was only administered because it helped the animals grow more quickly, and that allowed the farmers to get these animals to market more quickly. Now, the reason I bring it up is because it is no longer legal for farmers to feed their animals tetracycline. So it's important to know that there are no meats that are being sold in this country that come from animals that are being fed antibiotics. That used to be true, but it is no longer true today. The last thing I want to discuss today is how we go about testing for sensitivity to antimicrobial drugs in the microbiology laboratory. There are testing systems available that are called disk diffusion testing systems, or also by the term Kirby-Bauer testing. You've actually already done a little bit of disk diffusion methods in the laboratory back when we were looking at the effectiveness of disinfectants. If you recall, you created a spread plate of bacteria. You grew a lawn of bacteria on an agar plate, and then you placed these discs covered with disinfectant onto the plate because you wanted to see if the disinfectant could inhibit bacterial growth. This disc diffusion method is also the way we test for sensitivity to antibiotics. The difference is that these discs are actually produced commercially. So the discs that are produced by these commercial companies are impregnated with different types of antimicrobial drugs. And in the laboratory, we make spread plates with samples that come from patients. So we create the spread plate on the agar plate, and then we place individual antimicrobial discs onto the plate, and then we incubate the plate. And what we're looking for is an inhibition of growth of the bacteria. So when the plates come out of the incubator, we use a ruler and we measure any zone of inhibition that we see. Any place where the bacterial cells were not able to grow, were not able to divide, and so a clear region shows up on the plate. Now, if you recall in our experiment with disinfectants, we were only able to make broad qualitative statements. We were only able to say either yes or no. The disinfectant could or could not inhibit growth. What makes antibiotic sensitivity testing different is that we can make much more quantitative statements about this. We can talk about whether a particular type of bacterium is very sensitive to the drug, completely resistant to the drug, or somewhere in between. And that's because these discs are standardized. Again, they're commercially produced. There's a known amount of antibiotic in each disc. And again, a very widely used testing system. So what you're looking at on this slide are two agar plates that are being used in antibiotic sensitivity testing. On this side, you can see a plate that is right side up, if you will. The lid has been removed. And you're looking at a plate. It doesn't look like it's got any bacteria on it yet. There's no bacteria growing here. But it's been spread with a sample. And the antimicrobial discs have been placed on the plate. Remember, we always put a control disc on these plates somewhere. We always put a disc that has no antimicrobial drug in it on the plate. And that's so we can control the experiment. We will compare everything to that control disc. The control disc should not inhibit growth, right? It has everything in it except an antimicrobial drug. So the bacteria should be able to grow right up alongside the control disc. What we're looking for in the other discs is whether or not the antibiotic or antimicrobial inhibits the growth of the bacterium. So we make these plates, we put them in the incubator, and then we get something like this. You can see that on the agar surface there's sort of a cloudy, foggy looking lawn of bacterial cells growing. And I think you can just make out that around some of these discs there's a clear zone. There's an area where bacteria were not able to grow. That's a zone of inhibition. Now on this slide, you can see a couple of different things related to disc diffusion testing. This is a container of these commercially available discs. You can buy these from several different manufacturers, and you can choose which antimicrobial drugs you want in the disc. So you can buy them sort of in bulk like this, or you can buy them in these very narrow tubes like this, where the discs are actually stacked inside the tube. And the reason you might want to do that, why you might want to buy it in this form, is because you can take this tube and you can insert it into this device. This is a very handy little low-tech device that is used to place several different discs onto an agar plate at once, so you don't have to do it by hand. You load these tubes into this device, and all you have to do is place it over the agar plate and push down on the plunger, and it will release one disc from each of these tubes. And you get a nice, repeatable, easy to produce antibiotic sensitivity plate. And again, when the plate comes out of the incubator, you have to measure any zone of inhibition, because we're going to want to make a statement about whether or not this bacterium is sensitive or resistant or somewhere in between to each of these different drugs. So recall, if there's no zone around a disc, what's happened here is the bacteria were basically ignoring that disc. They were able to grow all the way up to the disc on the plate. So whatever's in the disc has no effect on that bacterial cell. If you see a zone, if you see a clear zone around the disc, that's telling us that the cells that were placed on the plate around this disc were not able to grow. They were not able to divide and grow. And that's because whatever's in the disc is interfering with their growth. It may even be killing the cells. When you take a plate out of the incubator, if you see zones, they're going to be of different sizes. You can really see that on this plate. You can see that for a couple of these discs, this one and this one, cells were able to grow right up to the edge of the disc, no problem. So whatever was in the disc didn't affect them at all. It's likely that one of these two is the control disc. And then you can see something like this, where there is a very narrow zone of inhibition. And this one too. There's a zone, there's a clear area, but it's quite narrow. And if you think about what that means, some of these cells were inhibited, for sure. That's what produced the clear zone. But cells were able to get pretty close to that disc before they were affected. In comparison, if you see something like this, a really wide zone of inhibition, that means that cells were affected way out to that area, all across that clear area. No cell was able to divide and grow. Whatever's in the disc is inhibiting that growth, or maybe killing the cells outright. I also want to point out that sometimes you'll see a result like this on a plate. This is related to the antibiotic that's in the disc, or the antimicrobial that's in the disc. Some of these drugs, sort of famously tetracycline is one of these, they just stain the agar. It doesn't mean anything beyond that. You can see that there is a zone of inhibition here. You would just measure this one just like you would measure all the rest. Now remember, when we measure a zone of inhibition, we measure across the diameter. You can see that down here in this image. You take your measurement with a ruler across the whole diameter of the zone, and we measure these zones in millimeters. And you record your results so that you can go to one of the published standardized charts for these antimicrobial drugs. And that's what we're looking at on this last slide. You can find these charts in any microbiology textbook, The makers of antimicrobial discs provide these charts to their customers. You can find them in any clinical laboratory that's doing antibiotic sensitivity testing. All you have in this chart is the information that you need to determine whether or not a particular bacterium is sensitive to a drug, resistant to a drug, or somewhere in the middle. So yes, there are certain bacterial species and strains where they're not quite resistant to it. In other words, they are affected by it somewhat, but they're not affected in a way that we would hope to see. So the clinician has to make a decision based on these results as to whether or not to use that antibiotic against that particular bacterium. So in the first column here, you can see a list of antibiotics, some of the more commonly used antibiotics. In parentheses, next to the name of the drug, is the amount of antibiotic that's in each one of the discs. So for example, for amoxicillin, each little disc contains 30 micrograms of amoxicillin, and this dosing, this concentration per disc, is determined through laboratory testing, through earlier laboratory testing, so that we can use that disc to determine antibiotic sensitivity. And then over here, these are the results. This is the information that we apply our measurements to. You can see these are measurements that would tell us that a bacterium is sensitive. These are the measurements that would tell us that it is intermediate, or what you might think of as moderately sensitive. Not really sensitive, but a little bit sensitive. And then over here, these are the measurements that would tell you that a bacterium is resistant to an antibiotic. So let's take a look at one. Let's say that one of the discs on a plate that you made contained erythromycin. And when you measured the zone of inhibition, you measured it at 17 millimeters. So what the chart tells us is that the zone would have to be greater than or equal to 23 millimeters in order for us to say that that bacterium is sensitive to erythromycin. In other words, that bacterium will not grow or will die outright if the patient takes erythromycin. So our measurement was 17 millimeters, so it doesn't meet this criteria. Our bacterium is not sensitive to erythromycin. Now let's look at the moderately sensitive column, 14 to 22 millimeters. We fit right in there. 17 millimeters was our measurement. So we can say that the bacterium that we're testing is moderately sensitive to this drug. It's not sensitive to the point that we would feel very good prescribing erythromycin for this patient, but there is some sensitivity to erythromycin. Over here in the last column, you can see that any measurement less than or equal to 13 millimeters would be an indication of resistance. So it's important to note that when a bacterium is resistant to an antibiotic, it doesn't mean that the antibiotic has absolutely no effect. This is still a zone of inhibition. If we had measured a zone of, let's say, 10 millimeters or 7 millimeters, it's still a zone of inhibition. There was still some inhibition of growth on that plate with that antibiotic. But through, again, other research, through other testing, we know that that small of a zone tells us that this drug is just not going to be effective, that this particular bacterium is not sensitive enough to erythromycin to make erythromycin a good choice. So anything less than 13 millimeters, and we're going to say, nope, this bacterium is essentially resistant to erythromycin. We're not going to prescribe it. So ideally, a physician wants to see results in the sensitive column. The more sensitive a bacterium is to an antibiotic, the more likely that antibiotic is going to be effective in clearing the infection. Sometimes we have to prescribe antibiotics that are in this moderately sensitive zone because that's all we have. But we try not to prescribe an antibiotic if the zone of inhibition falls in the resistant column.